So we talked a little bit last time about how plant cells divide and that these stages of the cell cycle are the same for plants as they are for any other eukaryotic cell. So just to review, they still go through mitosis, they divide cells. At this point, at the end of cell division, they're going to be forming something called a cell plate, which is a precursor to a new cell wall. But then G1, they would be doing their normal metabolism. They would be copying DNA and other cell materials during S phase. In this phase, G2, they might be elongating an existing cell there. So we're going to talk about, between today and next time, how do they build a new cell wall at the end of mitosis, and how do they elongate an existing cell that they have. This slide I'm going to skip over for this year, so just draw a slash through that. So we had talked about also last time looking at an overview of cell division for plants. And prophase, metaphase, anaphase, telophase are not particularly special to plants. So I'm going to assume that you got that knowledge from other classes. We're going to focus on this step and then these two steps. So before cell division starts, how do cells set where the plane of cell division is going to be? Where do they mark the boundaries for their new cell wall? And how do they assemble that wall to allow division of the cytoplasm after the cell divides? So pre-prophase and cytokinesis are where we're going to spend most of our time today. So one thing that plant cells do differently is because they're going to build a new cell wall across an existing cell, they actually mark where that's going to be really early on, before division even starts. And they do that by doing this process of building a pre-prophase band. A pre-prophase band. And this picture is showing fluorescent microtubules that have been labeled to show where this pre-prophase band is forming. So maybe that's a little bit more visible. And so this is a ring of microtubules that's forming around the inside axis of the cell. So that pre-prophase band is made out of microtubules. Darker marker here. It's made from microtubules. And it forms in a place that is depleted of actin. So this is a view of a cell like a box. This is the pre-prophase band of microtubules. And where that's forming is in a place where actin has been depleted. The actin depleted zone. So most of the time in a plant cell, and I'm going to pull this up a little bit to show this, in a plant cell and in other eukaryotic cells, there are this network of actin, and it would be in 3D space, that's totally underlying the plasma membrane. And in animal cells, that actin cortex, that can also be called the cortex of the cell, or the actin cortex, is providing support to hold up or hold out the plasma membrane so that the cell doesn't collapse. And plants have that too, and that's what these green ribbons are showing on here, is the actin cortex. And most of the time, the actin cortex is all over the cell. So sort of right under the plasma membrane, and it would be on the other side of the membrane as the cell wall. So when plant cells get ready to divide, something causes the actin in that network or in that cortex to be depleted or diminished in the place where the cell later is going to divide. And where actin is diminished, microtubules build up. How the actin diminishes and how the microtubules build up are still an open question. But microtubules will form this pre-prophase band in the place where cell division is going to happen later. So this structure is an early marker for the cell division plane. And wherever this is, that is where the wall is going to be later. There's quite a bit of time gap between setting this up and actually building the wall, but that's where the plant cell is going to build the wall. So in between here and here, we have mitosis. So prophase, metaphase, anaphase, telophase. That all proceeds similarly in plants. And then at the beginning of cytokinesis, the cell's not going to pinch together, it's going to form a new wall. And the initial wall that it forms is called the cell plate. So the cell plate is the beginning of a new wall. We call that the cell plate. And the cell plate, when it gets finished, then it's just cell wall. Again, one thing that I like in this section of the class is the book that I used to use has mutants for like every cellular process. And this particular mutant is called a FAS, or FOS mutant. And that is derived from a German word that means barrel. So probably the people that discovered this were German researchers, and they named the mutant after its phenotype. So barrel in German is FOS, and the mutant of this particular gene has a barrel-shaped phenotype. So we might label on here, this is the FOS mutant. It might be FAS, I feel like it's FOS. If you know German, I'm so sorry. This is wild type of an Arabidopsis seedling. So this is what normal seedling development should look like. And this would have been the radical end, epicaudal, plumule end. Here's the cotyledons, or the seed. It's probably the seed coat. This is the seed coat. These are the cotyledons. This is actually the plumule here. So this would be normal photomorphogenic development. And the FOS mutant has all the parts too. It's got radical, epicaudal, plumule end. There's the seed coat. It's germinated, and it's grown for the same length of time as that. It's got a problem, for sure. So the reason that it doesn't look like a normal seedling is that what the FOS gene controls, so its gene product controls the pre-prophase band formation. Something about that gene product tells the plant where it's supposed to make a pre-prophase band. And normally, let you finish writing that. Normally, the pre-prophase band is supposed to form around the middle of the long axis of the cell. I'm going to roll this down and go back. So a FOS mutant would not necessarily form the pre-prophase band here. It might form it up here. It might form it in a completely different axis. Cells are dividing, but they are not dividing in the appropriate way for a plant. So if you look back at this, and we imagine a cell in the epicaudal is a long box like that, they ought to be dividing in that direction. So form new cells and also expand them in that direction. And that requires perpendicular preprophase band to the axis of growth. FOS mutants form preprophase bands just wherever. It's unregulated. And they are dividing cells. So they are dividing, and those cells are elongating, but they're going in random directions. So that's why it looks like it's too fat, it's not stretching out. It's still got all the parts, but it's dividing in a inappropriate way. And its particular problem is that it doesn't form preprophase bands. Actually, you can maintain these. You can keep on growing this plant if you give it supplemental sugars. Otherwise, it will never make it beyond the seedling stage. Too wimpy. So, assuming that a cell can set its preprophase bands, and it marks out the correct place to build a new cell wall, plants build their cell wall in a very strange way when they're building a new dividing wall. And they build it from the outside, from the inside out. So that's worth noting here. New cell walls, which we call a cell plate. So a cell plate is like an unfinished wall. It would be like a scaffold wall. Cell plates, those are constructed from inside to outside. Or we could say interior to exterior. So if it were me and I were going to build a new wall across the cell, I would start from an existing strong wall. And we talked about that for this class. If we were going to build a new wall in here, you start from an existing wall and you go over. Plant cells start from the middle and build out to connect to the side walls. Very weird. And one reason why they do this is because they actually are going to use the spindle microtubules. They use spindle microtubules as a scaffold, or alternatively as a highway for the building materials. So if we look back at this cartoon, I'm going to go back a few slides. This cell now has 2 nuclei but no cell wall yet. It's binucleate. It's done mitosis, it's done telophase where it's separated these. But to get to this point, it had to build this network of microtubules to pull chromosomes. The spindle, remember that? The spindle is made out of microtubules. And the spindle, toward the end of telophase, is shown in the green here. The plant cell uses the existing spindle to send wall materials to the middle of the cell. So you've already got this big structure that exists and that can carry materials from place to place. That's why the plant cell builds wall from the inside out, because it's already got spindles, microtubules going to that spot. And that's shown here. So in telophase, toward the end of telophase, you've got these existing microtubules, and they are longer than that. They're not shown here, but they do extend across the cell. And you also have a bunch of vesicles at that place. So little membrane-bound vesicles that have arrived on these spindles at the middle of the cell. And those vesicles are going to fuse and start forming cell plate. Yeah.
Does all this happen like on top or underneath of the vacuole, or does
The vacuole, that's a good question. Does it happen on top or underneath or through the middle of the vacuole? I think the vacuole has to, I'm not positive about this, but I think the vacuole has to be taken down before this happens.
I was just wondering because there's a lot going on.
Because if not, it would be destroyed by all of this coming through it anyway. So vacuoles can help an existing cell expand, but during cell division, if you remember when we looked at like the growing part of a cell where it's dividing, and there were a lot of little cells that were not as boxy yet and they were smaller, it's easier for them to divide than a whole giant vacuole-filled cell. That's a good question. So this assemblage of microtubules and vesicles, and the contents of the vesicles, that's phragmoplast. And that's one of my favorite plant words, fairly bizarre word, phragmoplast. But its name kind of helps me remember what it is. So plast, what does plast tell you about the structure? Chloroplast, tonoplast, protoplast. Circle? They tend to be circular if they don't have a cell wall in with them. What did you say?
Oval?
Oval. Why do they form circles? That's related to the plastness. Plast implies membrane, like something being enclosed in a membrane. So phragmoplast is membranes coming from vesicles associated also with microtubules. So it's an assemblage of building materials and construction needs where the cell plate and the cell wall are going to be. So it's got microtubules in it, it's got vesicles in it, which are membrane-bound tiny compartments that can move along microtubules. So the vesicles have moved from the poles of the cell to the interior, and they are carrying wall cargo in them. A phragmoplast is like a construction site. It doesn't look good yet. It looks like a mess. But it is in the location and it has the materials that you need to build. That's phragmoplast. So a phragmoplast, once it gets further along, will become a cell plate, and a cell plate eventually will become a cell wall. It's sort of a intermediate stages. So this is a particularly nice picture of what a phragmoplast looks like in process. And what you're seeing here is this is a computer model in which someone tagged vesicles with fluorescent dye and they tracked their movement along microtubules. So these are tracks, it's almost like a time lapse. These are vesicle tracks where they went along microtubules and they came to the middle of the cell, to the place where the pre-prophase band specified a while ago. And they are fusing together here. So when you see these colorful dots, those are vesicles that haven't made it yet. Once they get here and they start fusing, they're colored yellow. The reason that the vesicles come here is that they move toward the plus end of microtubules. If you haven't covered plus minus end, that's a cell biology topic, and we'll get there in about a month. But microtubules have a polarity to them. So you can travel minus is usually at the start of the microtubule over to plus, which is at the end. So the microtubules plus ends are meeting here where the pre-prophase band set this axis, and vesicles are arriving here and then they actually are so crowded they start to fuse, and they are carrying cell wall components to that location. And they start to fuse in the middle of the cell and then spread outward in this phragmoplast. So this view and all the other views that we've seen so far, they're looking like from the side on. This picture that we're going to look at next, here's the side view, but here's what it would look like top down. So if we were to look from this direction, it would look like this. So looking through the long axis or looking down the long axis. And so this lighter orange, you can imagine, okay, there's some vesicles fusing. They're dumping material here to form the new cell plate, and it expands from the middle out. And it also matures from the middle out. So this is going to spread out this way, kind of in a, not a wave, but from interior to exterior. And so what's right at the middle is the oldest, so to speak. And it also cures, matures, solidifies from the middle. So this is now, you can almost think of this as phragmoplast, and the dark orange as cell plate. So cell plate starts to be finished and strengthened from the inside out, inside out, inside out, until you have a finished cell wall stretching across the correct axis of the cell. You see microtubules also disappearing from the inside out. So they're shown in the red. They're not needed anymore once you have cell plate forming. It's got enough material, it has to be strengthened. So microtubules also proceed from inside out until they disappear. And you can build this new wall. Okay, so next time we're going to talk about, okay, how is the wall actually structured? So we looked at how it gets built. We're going to look at components, how the components are linked, and how a cell wall can stretch for plants next time. We could turn that off.